Now, as we get to translation, I want you to realize how DNA stores information. DNA and mRNA by extension, because they're both doing it the same general way, are going to store things 3 bases at a time. We call this a triplet code, or we call it a codon. So when you've got your sequence of nucleotides here, and this is just showing an mRNA, we're going to look at this, we're going to read it in threes, these codons. So codon 1 will be AUG here, so an adenine, a uracil, and a guanine. And then you've got the next group of 3, the next codon, the next, the next, the next. And a gene can be hundreds of codons long. You know, in some cases, maybe even thousands. So these genes are going to be lots of nucleotides. But however many nucleotides there are, you've got to remember that you're breaking it up into like these 3 base words. And so even though there's lots of letters that are down there, there's actually not as much information being given as you would think, because you've got to break all these individual letters, these nitrogen bases, into threes, and each codon is going to code for an amino acid. So AUG will code for a specific amino acid, in this case methionine. You don't have to memorize that. There'll be charts we go over. You know, when you look at each of these guys, it's going to let us know what amino acid it codes for. And so we can then convert all these codons, this sequence of codons, into a sequence of amino acids. A sequence of amino acid is a polypeptide, or if it gets to be a bit more elaborate, we call it a protein. And that's what we're after, right? Because that's what gives us our phenotypes. That's what allows us to do the things that we need to do to be alive. That's how this code stores that information. So in translation, we're going to have where in the cytoplasm, we're going to use the code that is stored in mRNA, and we're going to use tRNAs, which are going to convert the code for us. So what that means is the tRNAs will come attached to a particular codon that's on mRNA, and they'll do that because they've got a little region called the anticodon, the complement. So in other words, if we've got on an mRNA AAA, there's a tRNA that has a particular region that binds to that would be UUU, the opposite, the complement. And so it would then attach to this, and when it attaches to this, it will drop off an amino acid that's also attached to the tRNA. So tRNA has this anticodon on one side that attaches to the mRNA's codons, and on the other side, it's got an amino acid. And so that's why we need it, because it's the one that can let us convert from codons to amino acids. And then you'd have the next codon on the mRNA, which could be CCC. And then you'd have another tRNA that has the anticodon for that, in this case GGG, would also have a particular amino acid. GGG will always code for a particular amino acid, and so it would then attach and drop off its amino acid. And so this would just go on and on and on until eventually we've got this long chain of amino acids that is our polypeptide that becomes our protein. Now, to facilitate this, though, we have to read the mRNA just right. Just like when you have a book, you can't just put spaces wherever you want. You don't want to put them in the middle of a word. And so the rRNA, the ribosome, is going to go through, and it's going to have these 3 sites, so it exposes one codon at a time. So the exposed codon will be at what's called the active site, the A site, and so that allows tRNA to bind there, the appropriate complementary tRNA. We'll then have a P site, which is where the guy that binds to the active site moves to, so he'll shift. And the P site is where we dump all the amino acids. So to help illustrate what's going on here, if we're looking at this particular image, the A site is going to be right here. So you're seeing the A site, this is the exposed codon, and this is the anticodon on a tRNA that's going to attach. And notice it has a particular amino acid attached to it. The P site is where the guy is holding all the amino acids that we've attached thus far. In this case, it's only 2. And the E site is where the tRNA is going to leave, you can see. So this is the E site here. And notice that it doesn't have any amino acids because it already dumped its amino acids on this guy. And so now this guy will attach, he'll move over. This guy will then move over, but before he does, he's going to dump all these amino acids onto him. And then the next guy is going to come in, and this process just keeps going, so whoever's at the P site keeps getting dumped with all these particular amino acids, and so you just keep getting a longer and longer chain of amino acids until eventually you're done, until eventually you reach what's known as the stop codon, because you have a start codon, AUG, that starts this process, and you have a stop codon that ends it. So this will go from the start to the stop codon, and then everything just detaches. All the tRNAs, the ribosomes, everything breaks apart, and what we're left with is a chain of amino acids that'll become a polypeptide or a protein, and then we have a bunch of tRNAs that go find a new amino acid so they can do this process all over again. And we've got a ribosome that's going to go try to find a new mRNA so it can attach to it and do translation all over again. And that particular mRNA that we're using can be attached to again by a different ribosome so it can keep getting used. Until this mRNA gets broken down by something chemically, it will keep being translated. So we can make many, many proteins from one mRNA that we produced. So now to understand a little bit more about how this works exactly, you should be able to read the chart that illustrates what's happening in translation. So what this is doing is this is giving us the mRNA is what this is describing. These are the codons. And so for each of these, it lists what amino acid that would particularly call for. So for instance, if I have UUU, that would be the mRNA sequence that would trigger a tRNA molecule that would be AAA, the anticodon, that contains phenylalanine, this is the particular amino acid, to come over and attach. So if I'm trying to figure out what I'm going to get in terms of the protein, I could say, well, UUU would then give me phenylalanine, which I'm going to put PHE just to keep it simple. And then let's say I had CCU. So if my next codon was CCU, it would trigger a tRNA molecule that would be, in this case, GGA, that's the complement of all of it, and that would bring a proline, so PRO, so that would be the amino acid there, and I'm going to draw these bonds because they'd get stuck together. Remember on that polypeptide, they connected them. And so let's say the next thing that we have is AGA, that's arginine. So then we'd have AGA could be our next codon, which would be, the anticodon would be UCU. And so that would bring arginine, so that tRNA would bring that. And then eventually we're going to have to get to a point where we stop, but I'm going to do one more and then quit. So why don't we just use an actual stop codon? You'll notice it's colored differently. There's only one start codon, AUG, but there's 3 stop ones. So then we'll just say we have UAA, which just makes it stop. So if this was the particular gene I have, which is tiny, this is not going to likely be a real gene. It's way too small. But just for illustrative purposes, you can see I can use this chart, which I would give you. This is not something you have to memorize, but you should be able to use this chart if I give it to you so that you could come up with the fact that this would build this particular sequence of amino acids. That's what you need to be capable of doing. You also should be able to, if I give you something like UAA and tell you this is mRNA, you should be able to realize that tRNA is going to be the RNA complement. So anywhere there's a U, it'll bind to an A. Anywhere there's an A, it would bind to a U, right? Because we normally would use T if it was DNA, but in RNA we use U instead. If you were talking about something that's GCG, then it would be the opposite for tRNA would be CGC, the opposite. Okay? If I asked you to convert something to DNA, so if GCG is my mRNA here, okay, the DNA version would be the same, right? CGC. There's no difference here between the tRNA and that. The only time you'll notice a difference is when we get tricky, where if I've got something like AUU, if we assume this is mRNA, then in DNA, remember that A will bind to T, and then U would bind to an A. That's the typical partnerships. So you should be comfortable if I ask you to convert stuff from DNA to mRNA, or if I ask you to convert stuff from mRNA to tRNA. You just got to make sure you realize what is the complementary partner for each, and then make sure you fill it in. With DNA, you should always have thymines included. When you're talking about RNA, you should always see uracils. It's an easy way to identify what we're looking at. If it's got a U, it's got to be some type of RNA. If it's got a T, it's got to be some type of DNA. And now lastly, when we first started looking at genes, we weren't quite sure how everything worked. So the first guys to really do much with this was Beadle and Tatum, and they were able to realize that when you mess with genes, they mutated certain types of bacteria, and they were able to show that when they mutated the bacteria, they could make it where they could not properly digest certain types of nutrients. And they could specifically figure out that these nutrients required multiple steps to digest. So essentially, A got broken down into B, which got broken down into C, which got broken down into D. So the actual organism needed D. Like you could give the organism D directly, that particular nutrient, and it was fine. It always lived. But if you gave this organism, let's say nutrient A, and you mutated it so you broke an enzyme, so we damaged like one of these guys here, you would see that the organisms would die because they could convert A to B, but they couldn't convert B to C. They could convert C to D, but they couldn't convert B to C to let them do that. And so by using this strategy, they were able to realize that when they caused a mutation, they were damaging one enzyme. And so this came up with this idea that one gene codes for one enzyme. And this was really the first time we were starting to plan out what exactly a gene was capable of doing. Now, later on, we've been able to do more research, so eventually this became one gene means one protein, because we now know that not all proteins are enzymes. They can also be pigments, they can also be chemical signals that we have. And so later on, we eventually refined this to the point that one gene codes for one polypeptide. And what a polypeptide is either a protein or part of a protein, because proteins are made up of one or more polypeptides. And so there are some particular proteins that might need multiple genes to assemble themselves because they're made up of 4 different polypeptides. And so that's why we don't say one gene, one protein. It's technically one gene, one polypeptide. But for the most part, with as simple as we're doing stuff right now, you can think that a polypeptide is roughly equivalent to a protein. I don't want to say it is and have you say I lied to you, but for our purposes, we're not going to go so in depth that that'll be a big deal. But later on, if you continue in biology, you'll see it does matter that it doesn't actually code for one protein, but it does code for at least a part of one protein, a polypeptide.